This method is a useful tool to introduce students to the concepts governing enzyme reactions. The main advantage of this technique is that it is suitable for students with very little wet lab experience, providing robust results for the students to observe and to interpret.
To begin the protocol, label a 15 milliliter tube suspension. Crush one lactase tablet containing 200 milligrams of enzyme into an even powder using a mortar and pestle. Place the resulting powder into the labeled tube and resuspend it in 10 milliliters of 100 millimolar phosphate buffered saline or PBS. Vortex the solution for 1 minute to maximize the enzyme extraction. Next, transfer 1 milliliter from the suspension to a 1.5 milliliter tube and spin the sample for 1 minute at 10,000 times G to sediment the solid particles. Transfer 500 microliters of the supernatant to a clean 1.5 milliliter tube labeled lactase extract. Place 390 microliters of 100 millimolar PBS into a 1.5 milliliter tube labeled reaction A. Then, add 100 microliters of 5 millimolar ONPG solution and mix well by vortexing. Add 10 microliters of extract into the reaction A tube and vortex the contents. Observe the reaction mixture often for 5 minutes and note any colorimetric changes of the solution. Set up